So we've developed this model of the arcuate nucleus as a place where convergent signals of body fat and gut signals regulate appetite and energy expenditure. We've also, in work I'm not showing you today, described how GLP-1 acts on this circuit, and that's been followed up by a couple of European investigators recently, and as well as how serotonergic compounds, in particular the serotonin 2C receptor agonists, act on this circuit, as well as opioids and antidepressants. What I'm going to talk to you now is about what goes wrong in obesity and how this circuit stops responding to signals, and potentially how we can bypass that, and how procedures like bariatric surgery might bypass some of that hormone resistance to reactivate this circuit. The final thing I'm going to talk to you about is some recent exciting data where we've shown how this circuit is also sensing glucose levels in the blood, and how when this circuit becomes resistant to blood glucose levels, we get increased systemic glucose in the animals. It's as if this circuit is a parallel system for regulating blood glucose. I think it's an important focus that it's worthy considering when we talk about the rates of diabetes remission we see after gastric bypass surgery. So in this paper here, we looked at what goes wrong in obesity. And in particular, we know that as animals or humans become obese, leptin levels rise. It's not as if obesity is associated with leptin insufficiency. Rather, it's a state of leptin resistance, analogous to insulin resistance in type 2 diabetes. These are studies from my lab looking at both fat levels in mice and a clinical study we did with Christian Roth looking at fat levels in humans. And the data is easily to understand. As you get bigger, leptin levels increase. It's not as if there's a failure of adipose tissue to produce leptin. We elaborated a model of diet-induced obesity in mice where we've discovered there are sort of 3 phases to the onset of obesity in animals. In the early phases, if we inject leptin in the periphery, the animals decrease their food intake. In the middle phases of the disease, after about 8 weeks on a very high-fat diet, animals don't respond to peripheral leptin injections, but if we inject directly into the brain, we do get a decrease in food intake. So the animals are retaining cellular leptin responsiveness, but there might be a defect in transporting leptin to the brain. In the final stages of the disease, leptin has no effect whether it's injected systemically or into the brain directly, and so we talk about this as a cellular leptin resistance. In this model, and you can see here an example of the size of these animals, after 20 weeks on a high-fat diet, these animals, the control animals, remain lean. Some of them remain lean on a high-fat diet, but the majority become obese on a high-fat diet, and that obesity is shown as increased leptin levels as well. What we found when we looked at hormone release from the brain is that the animals are resistant to leptin in so much as leptin no longer regulates NPY secretion. It's failing to suppress the feeding stimulator NPY. It's failing to suppress the feeding stimulator AGRP. And it's failing to activate alpha-MSH, the anorexic agent. And so these brains are no longer sensing how fat the animal is. We think this is because of increased levels of a molecule called SOX3, or suppressor of cytokine signaling 3. SOX3 is a negative regulator of leptin signaling, and it's also induced by inflammation. It's a cytokine pathway molecule. What we find here is that in lean animals, when we inject leptin, we get an increase in SOX3, but in the obese animals, there's a baseline elevation in SOX3, and leptin fails to mobilise it further. So it appears that the brains of these animals are in a state of chronic inflammation, and that's rendering them resistant to leptin because SOX3 blocks leptin signalling. So in summary, leptin electrically activates POMC neurons and inhibits NPY neurons, and that leads to increases in MSH secretion and decreases in AGRP and NPY secretion, setting up the brain for a negative feedback relay loop where high fat levels cause a suppression of food intake, causing weight loss. But in obesity, that's lost because of increases in this molecule called SOX3. And so the brains no longer detect how obese the animal is. We showed that, in work I'm not going to show you, that putting the animals on a low-fat diet and food restricting them can restore leptin sensitivity to this part of the brain. And I think this is potentially one of the mechanisms by which food restriction causes weight restoration. It's not just that we're decreasing food intake and having a caloric insufficiency, it's that we can reactivate leptin sensing in the brain and re-establish these melanocortin pathways as set point regulators. Obviously the big question is, can we bypass leptin resistance? And there's a considerable pharmaceutical effort underway to determine if that's the case. Now, that's describing fat sensing in these brains, but what about glucose sensing? Well, there's been a lot of work looking at how, in particular research in diabetes, how insulin sensitivity is regulated in the peripheral tissues and how insulin secretion from the pancreas is regulated. But there's been very little studied about how glucose is detected in the brain and what it means when neurons detect glucose. Probably the best described work on glucose sensing in the brain is around hypoglycemia unawareness, that is, how with repeated bouts of hypoglycemia, diabetics don't notice low blood glucose, and this is a serious consequence of recurrent insulin therapy. But it's not known what the neurons in the brain that are activated by glucose are doing. And in this paper a couple of years ago with Joel Elmquist and Brad Lyle, we described how these neurons are responsive to glucose, that is, they're more active at higher glucose concentrations in lean animals, and that they're doing this glucose sensing through a mechanism just like what happens in the beta cell. We also described how these neurons lose their glucose sensitivity in obesity, and if we engineer them to lose glucose sensitivity, the animals become diabetic. Finally, we describe a molecular pathway by which the neurons become glucose resistant, and show that if we block it, we've recently shown that if we block that molecular pathway, we can restore glucose sensitivity to animals and reverse the diabetes. So as I said, glucose sensing neurons have two modes of action, some that are active at higher glucose concentrations and then some that are activated by lowered glucose concentrations. The glucose-excited neurons use a beta cell-like mechanism. We don't really know how glucose is inhibiting glucose-inhibited neurons, and it's a subject of considerable research. This is a schematic of how a beta cell works, and it's analogous in the proopioid melanocortin neurons. Glucose enters the brain through through the cells through transporter, and then moves into the mitochondria where it undergoes oxidative phosphorylation to produce ATP, the cellular fuel. ATP moves to the membrane where it binds to a potassium channel, an ATP-sensitive potassium channel, and ATP closes this channel. That causes the cell to depolarize, and as the cell depolarizes, we get calcium entry through calcium channels that drives fusion of insulin-containing vesicles with the plasma membrane, causing insulin secretion. So in this way, we're linking extracellular glucose concentrations with the secretion of insulin. This is how the beta cell works. Because we knew that POMC neurons were excited by glucose, we expected a similar mechanism was happening in those neurons, and so we set out to molecularly investigate it. We first confirmed that the neurons were glucose sensitive, and what you see here is data showing the firing rate of neurons at 5 millimolar glucose and 3 millimolar glucose. These are relatively small shifts in blood glucose, analogous to what you'd see before a meal and after a meal. And you can see here a trace of the electrical firing frequency. You can see here at 5 millimolar glucose the activity. We switch it over to a lower glucose concentration. The glucose washes out of the tissue bath, and we get decreased frequency of action potentials, and then it's reversed when we add glucose back in. And this is the first derivative of that plot, the firing frequency. You can see a decrease in firing frequency that's sustained for the low glucose concentration, and then reverses when glucose is restored again. So these neurons are indeed glucose sensitive. And glucose stimulates release of alpha MSH from these neurons in the range that you'd expect in the brain after a meal. But we didn't know the physiological importance of it. It could just be that this is something that happens in these cells and has no real meaning. To determine how important it was, we created a transgenic animal with potassium channels that were unable to bind ATP. So we were trying to create an animal that was resistant to glucose levels in the POMC neurons. We used a technology developed by Chris Nichols at the University of Washington University. This is a technique whereby they block the normal effect of potassium on these channels, ATP on these channels. Normally, glucose metabolism increases ATP levels, which blocks the potassium conductance. And this happens through something called the KIR 6.2 pore-forming unit of the potassium channel. Chris mutated two residues in this ATP binding site on the channel and rendered it resistant to ATP levels. So ATP is not able to bind to the channel and therefore not able to close it. What you get is a failure of glucose to mediate closure of the potassium channel, and we end up with transgenic animals, at least when we put this mutation into the beta cell, that lack glucose sensing in beta cells, therefore they lack glucose-mediated insulin secretion and become diabetic. And we adopted exactly the same strategy, knocking this mutation into POMC cells. And what you see here is electrophysiological recordings of that effect. This is data just like I showed you before. In wild-type mice, there's activity at 5 millimolar and greatly reduced activity at 3 millimolar glucose, shown here. Whereas in the transgenic mice, with this very long label, we see no effect of glucose to change cellular activity, and that's what you see here. So we've made the cells electrically resistant to glucose. We've also made the cells hormonally resistant, if you will. Here you see glucose stimulating MSH secretion. Here we see no effect of glucose on MSH secretion. So the cells are resistant to glucose.